purpose of this PowerPoint is to review the structure of the DNA molecule and how that molecule is copied in the process of replication. So here is the DNA double helix. You can think of the double helix as a ladder. So the sides of the ladder are the alternating sugar, phosphate, sugar, phosphate, sugar, phosphate. And there's two different sides that are connected across by the nitrogen bases. Adenine goes with thymine and guanine always goes with cytosine. So that's what you can see over here. Phosphate, sugar, phosphate, sugar, phosphate, sugar. And then this side is upside down. So we say that the two strands run anti-parallel. That's the only way that the nitrogen bases will match up together. So adenine goes with thymine, cytosine goes with guanine, all the way up the entire strand. Thymine and adenine are joined together with hydrogen bonds, as are guanine and cytosine. So relatively weak bonds, but with all of them working together, it makes DNA a pretty stable molecule. We can label the ends of the DNA molecule. We say that the 5' difference between the 5' end and the 3' end. Again, the ends are opposite from each other since one strand is right side up and then the other strand is upside down, and that's the only way that the DNA molecule will copy this. DNA separate, and each strand with T and G is always going to join with C. Replication begins at specific sequences in the DNA called origins of replication. So it's not random. There are specific sequences of A and T and G and C that enzymes recognize as origins of replication. And an enzyme called helicase comes in and unwinds the DNA in both directions. And it forms a structure called a replication bubble. Okay? And this replication bubble extends in both directions, and it's within the replication bubble that the new strand of DNA is going to start to be created. So the helicase enzyme is going to separate the strands, and then another enzyme called DNA polymerase comes in to add the free nucleotides. So there are unattached A's and T's and G's and C's just floating around in the nucleus, and DNA polymerase into the strand. So here's free nucleotide. The difference between the nucleotide and the nucleotides that are already attached has 3 phosphates instead of just 1. Remember we talked about a nucleated sugar and a nitrogen base. These have 3 phosphates. So what happens with the help of DNA polymerase is 2 of the phosphates are removed, and that releases energy, similar to how ATP releases energy when it loses a phosphate. The energy that's released allows this phosphate to join to the sugar above it, and that adds the next nucleotide. So that's the job of DNA polymerase. It adds the free nucleotides. There's a problem with DNA polymerase, though. It's a picky. The problem is that DNA polymerase will only add nucleotides to the 3' end. And since one strand of DNA is upside down, that the molecule isn't going to create the copy in the right way. However, we've come across a solution to get around this problem. Okay? The way in which we create those 2 new strands of DNA, one strand of DNA is made continuously. The other strand is made in sections called Okazaki fragments, and then another enzyme called DNA ligase comes in and fills in the gaps. So we're going to look at the picture here, and I'm going to explain what's happening. So here's part of the replication bubble, and the square is DNA polymerase, and the circle is DNA ligase. And so you can see that one strand is 5' to 3', and the other strand is upside down 3' to 5'. So one strand is made continuously. That's called the leading strand because it occurs faster, and it's made 5' to 3', DNA polymerase adding the free nucleotides. Now, the second one works backwards, and it actually starts not directly at the end. It starts a little ways down, and it makes a little section 5 to 3, and then the DNA polymerase dissociates and reattaches and makes another little section 5 to 3, and then it makes another little section 5 to 3, and it continues. So it's working backwards, but it's still moving in the same direction of the replication bubble as it expands. These little pieces that are made are called the lagging strands, and there's gaps in between the pieces, and DNA ligase comes in and fills in those gaps. So if we look at the second picture, again, replication is occurring overall to the left. We can see that fragments 1 and 2 have been put together, and now the DNA ligase is starting to put together fragment 3, and DNA polymerase is making fragment 4. So all of these little pieces are called the Okazaki fragments after the scientists who discovered them, and that's how we can make 2 new strands even though DNA polymerase is picky and only works in one direction. Overall, we refer to DNA replication as semi-conservative. Each new molecule consists of 1 old strand and 1 new strand. So if it was completely conservative, the 2 old strands would wind back up together and the 2 new strands would wind back up together. But that's not what happens. We say it's semi-conservative because each new molecule is 1 old and 1 new strand wound up, and then 1 old and new strand wound up a second time. So replication is semi-conservative. It is possible through the process of replication that mistakes can occur. DNA polymerase, before it moves on to add the next nucleotide, it does double check, and usually it finds the mistake immediately. So here you can see that there's a mistake. DNA ligase and DNA polymerase work to fix that mistake. So enzymes come in and cut out the piece of DNA that's incorrect. DNA polymerase goes back over and adds the correct piece, and then DNA ligase comes in and fills in the gaps. So DNA ligase is also really important in repairing DNA if it's not properly matched up with the first time through replication.